This is the 9th video of PreventSARS3 Project, and this video I've talked about previously 5 of the previous coronaviruses, and this is the genetically most related coronavirus, SARS coronavirus. I will talk about this, and the objective is just to understand how natural SARS coronavirus 2 and so let's talk about SARS. The SARS virus had 2 years of windows of infection. It's genetically most related virus, and its fatality was 10% in ordinary people, but it reached to 50% in fatality in elders. Unlike SARS coronavirus 2, which has superior in almost maybe all viruses, it had no sialic acid binding. It evaded that capacity during host human adaptation. And SARS coronavirus also tried to have some mutations on its spike protein receptor binding domain. It was binding ACE2 receptor, but failed to do that. And it's the genetically most related virus to SARS, and within the context of host adaptation, it's completely not like SARS coronavirus 2. So this is how normal coronaviruses adapt. Actually, cannot adapt. But this is the important part. Here you can see the italics part is the sialic acid binding domain had no function to bind sialic acid. But SARS tried to, but had a lot of mutations, as you can see on this domain, and tried to improve that probably function on these high frequency mutations. So it's completely different from its genetically related SARS coronavirus 2. So this is something very interesting because this bold letters are its receptor binding domain. SARS coronavirus 2 had relatively few less mutations in this domain, receptor binding domain. It virtually couldn't be able to adapt that receptor that effectively. Tried to do that thing, but some of those mutations caused the virus to lose its capacity to infect human cells. It vanished, it disappeared. But it's completely, in this sense, also converse. We're going to see a lot of mutation in this domain in SARS-2. So both SARS coronavirus and SARS coronavirus 2, they are both beta coronavirus lineage B members, but they have a lot of differences. But another major difference is SARS-2 gained the capacity to be cleaved by furin. SARS coronavirus did not have this trait. And not only that, no other genetically related virus in that area, in that group, does not have it, suggesting an artificial insert. And SARS infected almost 8,000 people in 2 years. So therefore, how natural SARS-2 could be with its pandemic capacity. So thanks for watching. I will keep on getting into, right now we will get into SARS-2 after this video. Thanks.